Neurons have very characteristic shapes. I love art, and these images of these neurons, particularly when you start staining them, and they're beautiful shapes. And then to look at how chemicals can change those structures of those neurons, and then zoom all the way back up to behavior, where I can actually look at an animal behaving. It's just really fascinating. So I want to set the premise for how we got to thinking about the lung in the context of neurodegenerative disease. It's now very well appreciated that it's an interaction between these genetic risk factors for neurological disease, as well as environmental factors that really determine your individual risk of disease and or the severity of that disease. And so my lab is really focused on identifying what are these environmental factors with a real focus on chemicals in the environment. But what do we know about air pollution actually having effects beyond the respiratory system? By protecting people from environmental risk factors, we can improve health on a large scale. Historically, we have not had much data or very good data on how chemicals affect the developing brain. We've developed 5 different programs, and one is very much focused on the developing brain. So we've been looking at how PCBs may increase risk for neurodevelopmental disorders. And then we've also done a lot of work looking at organophosphate pesticides and their effects on the developing brain as well. Another major project in the lab is looking at the effects of organophosphate pesticides on the adult brain, particularly mimicking occupational exposure. Another area of research has been looking at how organophosphorus pesticides change that interaction between the neuron and the respiratory smooth muscle to drive asthma. And then another area of research is the Counteract Center, which has become a very large focus in my laboratory. And this is a collaborative effort across 5-6 different labs here at UC Davis. We're very much focused on chemical threat agents. And this is a national program funded by the NIH. We're trying to figure out what are the mechanisms that are linking acute exposure to long-term effects to develop more effective medical countermeasures. Our most recent research project, which is pretty exciting, is the effects of traffic-related air pollution on both neurodevelopmental disorders, such as autism, but also on neurodegenerative disorders, such as Alzheimer's disease. One of the innovative aspects of our research has been that we really make a real true effort to try and recapitulate the human condition. We've built a vivarium next to a tunnel, a heavily trafficked tunnel, collecting air from that tunnel and exposing animals directly to it in real time. That was a project that really reflects the power of interdisciplinary research, bringing engineers together with toxicologists, with neurobiologists, to really think of this really novel way that we can capture human exposure.
One of the great innovations that Pam has brought forward is the collaboration between the biological scientists and the physical chemical scientists and engineers like me, who can then produce the atmospheres that are needed to expose the animals.
In order to really get enough information to be able to regulate chemicals for developmental neurotoxicity, we need to switch to new testing paradigms. We've actually been involved in the efforts to adapt the zebrafish as a model for screening or testing chemicals for developmental neurotoxicity. Our companion animals are living longer and longer and longer, and we're starting to see a lot of the same neurological diseases appearing in our companion animals that appear in humans. And so we can have a real impact on that human-animal bond by preserving brain function in our companion animals. Respiratory toxicology.